I'm happy to share that this year at ASH, Mayo Clinic will be sharing our assessment of myeloma patients who received CAR-T across all 3 sites at our institution. And these includes both the FDA-approved CAR-T as well as CAR-T therapy given on phase 2 and beyond clinical trials. So it's not specific to any product, but how we look at response in patients with myeloma from the MRD assessment standpoint. And I think some of our findings are also corroborated by other abstracts that are also presented from the registration studies, including CARTITUDE-1 and CARMA-1 this year. And what's an important thing that we're seeing is certainly serial sustained bone marrow MRD negative status has very important prognostic value. That's not unique to CAR-T. We've seen it with all other types of myeloma therapies. The question that does come up is, for patients who receive CAR-T, often they're staying at the CAR-T treatment center through month 1. That's the first time that they may be getting an assessment of their response, and then they're returning home. So the follow-up monitoring is really a collaboration between the CAR-T treatment center and the primary hematologist at home. So we wanted to look at, well, what's the significance of that? Can that provide some information? And I think the important finding is that the month 1 assessment, particularly if the patients achieved bone marrow MRD negative status, and we also added assessment for PET MRD status that does help increase some sensitivities, is these can be very reassuring. Now, we still need serial assessment to see if they are maintaining that MRD negativity, but the odds are quite good that the patients may do quite well and continue to be in response. However, for the patients who are MRD positive already in the bone marrow and/or with PET, that is regardless of the cellularity of the bone marrow at that time, these patients in general do not do well. We're not really seeing a significant number of these patients converting eventually to MRD negative status. None of these patients ever achieve CR or stringent CR. And so I think we're identifying that this is a high-risk patient population in which, at the minimum, they need to be monitored a lot closer. But where we want to do more testing is strategically design clinical trials to understand what type of early intervention treatment in this setting could perhaps help these patients. And there are other investigators in the myeloma field this year presenting data from ASH, including results, some of the analysis from the correlative samples on the clinical trial, finding that these early clones of refractory residual myeloma cells may be quite different from what we may see later, MRD positive clone that might show up after the patients achieve their best response. And so I think some of these information can be used to help inform us how to design future clinical trials. I think the value of our study that's corroborated by also additional data from clinical trials is really identifying these high-risk patient populations to inform the next areas of investigation.